Good morning, students. Today we will be starting with the topic AIDS. AIDS stands for Acquired Immunodeficiency Syndrome, and it was for the first time reported in 1981, and since that day about 25 million people have died because of AIDS. AIDS, as it is acquired immunodeficiency syndrome, means it is acquired during the lifetime and is an immunodeficiency disease. Syndrome word means it is a collection of symptoms, collection of infections because of which the immune system becomes which is due to the immune system deficiency, and it is caused by a virus, human immunodeficiency virus. In short, we call it the XIV. This disease makes the immune system weak, and it becomes weaker and weaker over a period of time, and then the person is susceptible to a great number of diseases. The body becomes weak, the body loses its ability to fight infection, and therefore the person suffering from AIDS or a person who is HIV positive, human immunodeficiency virus positive, becomes weak and is susceptible to a variety of infections. The HIV virus is a retrovirus with RNA as its genetic material. And to make people aware of the disease, December 1 is celebrated as International AIDS Day, where people can be educated, made aware about AIDS, how it is spread, what is the prevention, what is the way in which people can protect themselves from this virus, because till date no cure has been found out for this disease. Now, mode of infection of AIDS or spread of it is the infection from HIV virus can occur firstly when a person has sexual contact with an infected person, then by sharing needles, as in case of drug abusers, exposure to infected body fluids or tissues, mainly blood, and from an infected mother to the fetus through the placenta. Now, HIV has also been found to be present in tears and saliva, but no instance of transmission from these body fluids has been reported. There is no danger of infection with AIDS virus, that is HIV, by casual social contact or by sharing objects or food. So we have discussed the basic things about AIDS. Full form of AIDS is acquired immunodeficiency syndrome. It is due to deficiency of the immune system, which is acquired during the lifetime of a person. It is caused by HIV, that is human immunodeficiency virus, which is a retrovirus with RNA as its genetic material. Discovered in 1981, the immune system becomes weaker and weaker, and a person can contract various diseases. The word syndrome in AIDS stands for collection of symptoms or collection of diseases. December 1, celebrated as AIDS Day, to create awareness and no cure has been found out till now. There is no cure till now. The retrovirus has RNA as its genetic material. Now, if we see the structure of HIV, we will see that there is a RNA molecule surrounded by a viral protein coat, and this is the RNA, which is the genetic material of HIV. Then we have the envelope made up of glycoprotein. Structure is like this. Just showing you one portion like this. This is the glycoprotein envelope. So this is basically single-stranded RNA, which is the genetic material. This is a retrovirus, HIV. Now, reproduction of this virus takes place when the virus infects a cell, and the RNA by reverse transcriptase produces a copy of the DNA. That DNA then incorporates into the nuclear DNA of the cell, of the animal cell, and prepares multiple copies. That's how new viruses are produced, and these new viruses then infect new cells. So if we see the life cycle now, if we see the life cycle of this virus of HIV, we will see this virus infects an animal cell. Suppose this is an animal cell. So the virus infects the animal cell. The animal cell has a nucleus. The nucleus has its own DNA. This is the chromatin network. This is the nucleus of the cell. So once it enters this, the viral RNA makes a copy of DNA. This is DNA. This DNA then incorporates into the nucleus of the cell, host cell, nucleus of host cell. Replication takes place. It gets incorporated. Replication of DNA of the host takes place. New viruses are produced. And these new viruses then infect more cells. So this is how the DNA of the virus replicates in the nucleus of the host cell, and more copies of the virus can be produced. HIV infects and kills the T helper cells, Depletion of the T helper cells weakens the immune response of the body. As I told you, HIV is a retrovirus that utilizes reverse transcriptase to produce a copy of the DNA. This is called cDNA, and the enzyme that produces DNA from RNA is reverse transcriptase enzyme. Reverse transcriptase enzyme produces cDNA, which integrates into the cell's chromosomes before it reproduces and makes copies of itself. HIV attaches itself and binds with the plasma membrane of the T helper cells. HIV enters the cell and uses a special enzyme called reverse transcriptase to make a copy of DNA of the genetic material. This single strand replicates and produces double strand cDNA. Now double-stranded viral DNA into the host cells, where it directs the production of viral RNA. Each strand of viral RNA brings about synthesis of the viral proteins, and the assembly of RNA strands and viral proteins gives rise to many viruses, and these viruses are then released from the host cell before it finally dies. The HIV formed attack more and more helper cells, replicate and produce more viruses. This is repeated and it leads to progressive decrease in the number of helper T cells, and that's how the immune system becomes deficient. T helper cells help in fighting infection and they are getting damaged. The damage is caused to the T helper cells, and because of which the immune system becomes deficient. And S syndrome, collection of infections, can be seen in a patient suffering from HIV, and that is when we call it AIDS. People infected with HIV may not show physical symptoms of illness for quite a long time. The period may vary from a few months to 5 to 10 years. That is called the incubation period till the symptoms are seen in a person. Once an individual is infected, the body may respond in many ways. Swollen lymph nodes and fever. Sometimes it may be persistent cough and fever associated with difficulty in breathing, decreased count of blood platelets, which leads to hemorrhage and fever, sweating at night and tiredness, weight loss and loss of appetite are the other symptoms. HIV may even attack the nervous system, causing damage to the brain. It may lead to memory loss, loss of coordination, partial paralysis, mental disorder, and loss of ability to speak and think. HIV destroys the protective immune system of the body, so the person becomes susceptible to other infections. The other germs like bacteria, fungi, protozoans, and even cancer cause to infect and destroy the body. Common diseases that are seen in HIV patients are pneumonia, tuberculosis, and certain kinds of cancers. Then we come to prevention. So far, there is no vaccine to prevent AIDS. It can only be prevented by bringing changes in personal behavior. One should use only disposable needles and syringes for testing of blood or for taking injections. Use of fresh, new blade at the barber shops. During any blood transfusion, blood transferred should always be tested for human immunodeficiency virus. Sexual contact with unknown people should be avoided. Regular check for HIV in susceptible populations should also be promoted. So these are some of the ways in which HIV can be prevented, because till date there is no vaccine, no cure for AIDS. Control. Treatment of AIDS with antiviral drugs is only partially effective as they can prolong the life of the patient. Means diseases that are caused because of the weakening of the immune system can be taken care of, but AIDS cannot be cured fully. Now, the diagnostic test for AIDS is called ELISA, that is enzyme-linked immunosorbent assay. The diagnostic test for AIDS is ELISA, which stands for enzyme linked immunosorbent assay. Now, ELISA is the most widely used test for AIDS. Usually, persons that are infected with HIV may show symptoms within 2 to 6 weeks of exposure. Sometimes, virus may remain silent for very long periods, for a period of up to 10 years also. When HIV enters the bloodstream, it begins to attack the white blood cells, the T lymphocytes, and the antibodies are produced in the body. These antibodies are tested and detected by a specific blood test, which is ELISA, usually 2 weeks to 3 months after infection. Now, procedure of this diagnostic test ELISA. ELISA, that test only detects the antibodies to HIV and not the virus itself. Once the virus has infected a person, it has attacked the lymphocytes and lymphocytes have produced antibodies against the virus. So, ELISA tests the presence of antibodies in the person suffering from AIDS or person who is HIV positive, but it does not detect the virus itself. The steps or procedure for this test are blood is taken out and the serum is separated. Serum of blood is applied to an ELISA plate on which unactivated HIV antigens are bound. Now the plate has antigens and serum is applied on this plate. You know there is antigen-antibody reaction. So if the patient is exposed to the virus, the antibodies will bind to the HIV on the slide. So if there are antibodies against HIV in the person, these antibodies in the blood of the person will bind to the antigens present on the plate, forming a complex. The bound antibodies can then be detected by an antibody enzyme solution. The sample is mixed with an antibody enzyme solution to which only HIV antibodies will attach. Chromogen, which is a substrate, is added which changes color when cleaved by the enzyme attached to the second antibody. Finally, the slide is treated with another enzyme, and if it changes color, it simply shows that HIV antibodies are present. If no HIV antibodies are present, there will not be any change of color in the patient blood sample. So this is how AIDS is detected. The virus itself cannot be detected, only the antibodies that have been produced in blood in response to HIV will be detected by this diagnostic test called ELISA. So, AIDS is a deadly disease. It is dreadful because the immune system gets destroyed over a period of time, and since there is no vaccine, no cure, a lot of deaths are caused because of AIDS. Therefore, prevention is the only option to protect oneself from AIDS. With this, we have completed the chapter Human Health and Diseases, and as I told you, for your half-yearly exams, questions from Unit 3 from this chapter will be there in the paper. The other two chapters of Unit 3 we will be doing after the half-yearly exams are over. So tomorrow I will be posting a practice assignment for you, in which I will include a few objective and a few short answer questions so that you get into the habit of writing the answers. I have already told you about the pattern of the paper, the syllabus. Good luck to all of you. Study well.